Hello everyone and welcome to my seminar presentation entitled General Insights on Microfluidics, the Role in Nanomaterial Synthesis and Diagnostic Hematology. In this presentation, we will discuss fundamentals of microfluidic devices, the materials that are used to create these devices, the function of these devices in the microfabrication and nanomaterial synthesis, and their applications in blood analysis as well as sweat chloride test. And finally, in a graphical abstract, we will summarize everything that we have discussed within the presentation. So the word microfluidics is the science of flow at the microscopic scale. It is not a completely foreign word to us, as we can see microfluidics everywhere around us in the natural environment, such as in the leaf capillaries, blood capillaries, and spider webs. But it is also a mature technology that is found on the e-readers, inject printers, and lab-on-a-chip devices. A brief history onto microfluidics. The whole thing started in the early 1950s with the development of photolithography, which happened to be one of the basic processes for the production of these microfluidic devices. One decade after that, miniaturized systems on silicon wafers were beginning to emerge. In the 1980s, the first generation micropumps and valves were established, and one decade after that, the concept of lab-on-a-chip and microreactors was also beginning to emerge. By the 2000s, polymer-based microchannels and multi-layer soft lithography were beginning to emerge, and these technologies helped to make microfluidics much more inexpensive. Currently, the microfluidics are involved within a different variety of fields, such as molecular biology, proteomics, cell biology, chemistry, theranostics, as well as point-of-care diagnostics. So why should we care that our microfluidic devices are small in size? Obviously, for the numerous advantages the small scale would provide them with, such as decreased material consumption, optimized interactions, reduction in the reaction time, reduction in the sample processing time, and having an overall lower cost. This is an example of a microchip. Now, microfluidic devices also require other components additional to the chip, such as integrated pumps and valves, electrical fields, electrodes, and electronics to become complete diagnostic systems. This network of trapped microchannels is connected to the outside by inlets and outlets that are punched through the chip as an interface between the micro and the macro world. The microscale behavior of microfluidics provide them with unique fluid dynamics, all in relation to the scaling law, which describes the variation of a physical property as a function of the size of the system. For instance, for larger objects, the gravity is very noticeable and has a great effect on the object. However, on the microscale microfluidic device, the gravity is pretty much negligible. Similarly, for larger objects, the volume is much more important than the surface forces. However, inversely with the microfluidics, the surface forces like the viscosity and the capillary forces become much more important. Now, since we're discussing microfluidics, we need to understand the type of fluids that go through the system. Generally, there are two types of fluids, Newtonian fluids and non-Newtonian fluids. The viscosity of the Newtonian fluids are independent on the shear stress, which means if I bring a beaker and place a Newtonian fluid inside, it doesn't matter how fast I stir that fluid or for how long, the viscosity will stay the same and it will never change. On the other hand, non-Newtonian fluids' viscosity is actually dependent on the shear stress. An example of Newtonian fluids would include the water and the gasoline. Examples on non-Newtonian fluids would include the toothpaste and the butter. For microfluidic systems, Newtonian fluids are to be utilized. Now that we know what types of fluids go through the system, we should also know the type of flow that it uses. And there are two types of flows, the laminar flow and the turbulent flow. The laminar flow travels smoothly or in regular paths, in contrast to the turbulent flow, in which the fluid undergoes irregular fluctuations and mixing. The The type of fluid that is usually used in microfluidics is the laminar flow. The laminar flow in microfluidics have several benefits. The fluid patterns in the laminar flow are much more predictable, much more easily controlled, and easily manipulated. It enables extremely high throughput processing while preserving the properties of every single unit. It also allows the transportation of matter through diffusional mixing, and the rate of transport follows the Fick's law of diffusion, which is proportional to the concentration gradient from high concentration to the low concentration. We move on to study the materials that make up the microfluidic device. The most common materials that are usually used include silicon and glass, ceramics, polymers, and composite materials made of organic and inorganic substances. Now, the reason why these are the most common materials used is due to their properties. For example, the silicon is abundant and resistant to organic solvents. However, it's costly and leaking, so perhaps it's not always the first option. The glass is available and biocompatible material. Ceramics have good electrical and mechanical properties, and polymers have attracted a lot of attention because they are inexpensive, easy to obtain, and are flexible for modifications. This is a video to show the microfluidics fabrication in the lab. After the video, I will break down its components in a simple diagram to help understand the process even more easily.
So here we go. We take a silicon wafer and pour resin on top. We spread it by spinning it very fast and heat it up to solidify the resin. Then we take the mask and put it in the lithography machine. We also put our wafer with the resin. We light it up, take the resin back out, heat it up, and develop it. We get rid of the non-lit up parts with a solvent. We clean it, and the mold is ready. We start by surrounding the mold with aluminum. We take liquid PDMS, then some crosslinker, mix them together, and pour the mixture on the mold. The whole setup is put under a vacuum belt to eliminate air bubbles, and then we wait for about 10 minutes. Once the bubbles are gone, we take the PDMS and put it in a heated enclosure to speed up crosslinking. After an hour, we can take out the PDMS, which has become solid. We take away the aluminum and the mold and cut out of the PDMS the area around the circuit we want. Now we just need to make holes with a punch for the liquid's inlets and outlets. We're going to put the PDMS chip and a glass slide in an atmosphere of extremely energetic pure dioxygen. It will make every exposed surface very reactive. All it takes is putting the PDMS on the slide for chemical bonds to form. We've just made an irreversible bonding.
This is a summary of what the video was discussing. The two processes used to fabricate microfluidics include photolithography, which was done in the orange room, and soft lithography process. In photolithography, a silicon base is used, and a resin called photoresist is poured on top. The photoresist is sensitive to the UV exposure and becomes insoluble upon reacting with UV. For that, we use a mask that has the negative of our channel circuit of interest, so when we expose it to the UV, the areas uncovered by the mask will become insoluble. Then, we do a washing step with a solvent called the developer to dissolve unsolidified resin. In the soft lithography, we utilize the mold we created earlier and cast PDMS to make replicas. Then, the PDMS is exposed to oxygen plasma treatment to make it surface reactive and place it on glass. Now our microfluidic chip is ready for use. We move on to discuss microfluidic functions in nanomaterial synthesis. Usually, microfluidic devices are utilized to synthesize nanomaterials used for theranostic applications. By theranostic, it means that the nanoparticle will have a diagnostic entity and a therapeutic entity all at once at the same time. The microfluidic reactors are advantageous for the nanomaterial synthesis because they enable precise reaction control, rapid chemical reactions, they allow for the flow synthesis of micro or nanoparticles, they are time efficient, cost saving, and a lot of examples of nanomaterials can be synthesized, like the gold and silver nanoparticles, magnetic nanoparticles, quantum dots, silica, hydrogels, and hybrid composites. The strengths of using microfluidics in nanomaterial synthesis involve the implementation of multiple stages for the particle production. For example, we're not only synthesizing the nanoparticles, but we are also doing quality assurance and monitoring as they are being synthesized in the system. Not only that, but even for quantum dots, the emission characteristics can be monitored upon the synthesis. However, there are some weaknesses in microfluidics nanomaterial synthesis involving the surface deposition or the clogging within the microfluidic channels. One way to resolve this problem is to do salinization and pH adjustment. Salinization is essentially covering the surface of the microfluidic channels with a coating that contains silane-like molecules to make the surface chemically inert and unreactive. However, surface deposition is not always bad. Sometimes we do it intentionally for real-time sensing and diagnostic applications. In a nutshell, our microfluidic system should have inlets for liquids to enter the system. These liquids can be mobilized into the system via a pump or a syringe. We also need to have channels to allow liquid circulation and interaction. And finally, there should be outlets to collect and analyze the fluids that we transformed. This video will show us the droplet-based microfluidic synthesis of nanoparticles. After that, we will take an example from the literature to compare the batch synthesis of nanoparticles versus the microfluidic synthesis.
One thing microfluidics is great at is making consistent droplets of fluids. They can be used in cosmetics, chemistry, biology, and even as medicine. To make droplets the regular way, take a beaker filled partly with water and partly with oil, two liquids that can never mix. When you stir, droplets start to form. They are of different sizes. When using this technique at an industrial level, inconsistent droplet size can be an issue, and some of the liquid will be lost because you cannot control the quantities precisely. Now, imagine being able to control the exact size and quantity of droplets with a faster and more precise process. Microfluidic equipment can do that. For the same experiment, you need a basic microfluidic chip, a see-through slate with engraved channels only a few nanometers wide. In the microfluidic channels, you can control how you make the droplets. Put the fluid for droplets in one channel. The other fluid will pinch it from both sides and make tiny drops of fluid that collect in the pool on the right. Their size is strictly controlled. There is no waste of product, and the process is fast. You can make up to 1,000 droplets per second. Great! But why make droplets at all? Well, this miniature droplet is actually a microreactor. It can be made of any type of fluid, and we can trap what we want to observe inside. This microreactor droplet can be used for many things: cosmetic creams and lotions, supramagnetic nanoparticles, diagnostic tests and assays, mayonnaise and low-fat food with air-filled fat particles, controlled medicine delivery with biodegradable microparticles, and many more.
This is an example on nanomaterial synthesis using microfluidics. In this study, the researchers compared the batch synthesis versus microfluidic nanomaterial synthesis via the nanoprecipitation method. For the microfluidic system, PLGA polymer in solvent and the drug was pushed through one channel, and then the perpendicular channels surfactant in water was added to produce the nanoparticles. These are the results of the experiment. We can clearly see that the batch synthesis resulted in inaccurate results because the sizes and the size distribution between every different experiment was different. However, when we compare it to the microfluidic synthesis, the size and the size distribution were predictable and accurate, which indicates the suitability of using microfluidic systems for nanomaterial synthesis. Next, we move on to the applications of microfluidics in blood analysis or hematology, where we will be discussing three components: troponin, glucose, and complete blood count (CBC). We start off with troponin, which is a widely accepted biomarker for cardiac injury. It helps regulate the skeletal and the cardiac muscle contractions and consists of three regulatory proteins: troponin T, troponin I, and troponin C. However, troponin T and I were found to be most sensitive and specific for the measurement of cardiac injury. This table presents an overview of existing point-of-care assays for the quantitative detection of troponin. We can see that the main technologies used are either lateral flow immunoassays or ELISA tests. And some of these technologies have integrated microfluidics within them. To take in the first example, the iStat CTNI is an ELISA test with integrated microfluidics. If we compare them with regular ELISA tests, we can see that the assay runtime of 7 minutes is much shorter than 17 minutes, 14 minutes, and the 20 minutes that don't have integrated microfluidics in them. So as a general refreshment, the ELISA test is the enzyme-linked immunosorbent assay. It's basically the kind of test that uses 96 well plates, and it also requires incubation periods of either several hours or overnight. Generally, in these tests, the 69 well plates have antigens that are coated on them. We wash them with the patient's serum, and if the patient's serum contains antibodies for these antigens, it will bind to them. Then, we make a second washing step, usually using an animal serum, that contains an enzyme-linked antibody that will link with the previous complex. Then, we do another washing step with a substrate solution, and the substrate will bind to the enzyme and gives us a calorimetric indication where a color indicates a positive test. The integrated microfluidics ELISA CTNI measures the cardiac troponin I using somewhat different components. For example, it contains a PDMS pump and valve to help push the fluids through the microchannels. It has a substrate solution preceded by a wash buffer solution, and at the tip of the chip we have a conjugate pad where we will insert our sample on, and right in the middle we have a reaction chamber which contains 96 wells. The assay is conducted in 3 simple steps. First, we dispense our sample solution onto the conjugate pad. The sample should have the antigens of interest that will bind to the detection antibodies that are found on the conjugate pad. Then, the antigen detection antibody complex will move through the microchannels and onto the reaction chamber to bind to the capture antibodies that are found in the 96 wells. So now we have a sandwich of detection antibody, antigen, and capture antibody. The wash buffer will move in next, followed by the substrate solution, which will generate perceivable color interacting with the enzyme-linked detection antibody. The benefits of using this system is that we are reducing the reagent consumption, reducing the sample volume because we are only using 30 microliters of blood. The assay time is shortened, and we have a highly sensitive detection where the limit of detection was significantly improved when compared to the conventional ELISA method. It is also an easy-to-use operation. These are the results of the research when they compared the proposed methods of microfluidic integrated ELISA versus the regular conventional ELISA. 2 observations can be seen from this graph. As we increase the concentration of the CTNI, the color intensity also increases, and we can also see that both of the methods were effective in measuring the concentrations of the CTNI, indicating the successfulness of using microfluidics within the ELISA system. Here is an example of microfluidics integrated ELISA device for troponin I measurement.
With the iStat blood chemistry analyzer, patient-side, lab-accurate blood tests are available in minutes. The iStat analyzer was designed with ease of use in mind. Performing a patient test requires just a few simple steps. First, scan the operator, patient, and iStat cartridge lot number codes. Next, fill the cartridge with fresh whole blood. Finally, insert the cartridge into the handheld. In as little as 2 minutes, you can view results, making the iStat analyzer one of the fastest methods for analyzing patient blood samples on the market. With the iStat analyzer, your facility can perform in-house diagnostic testing, obtain lab quality results within minutes, and accelerate clinical decision-making.
The second technology to measure the troponin levels, as indicated by the previous table, is by using lateral flow immunoassay. It is a paper-based microfluidic system composed of sample pad, conjugate release pad, nitrocellulose membrane, as well as an absorbent pad. The lateral flow immunoassay can work with a range of liquid samples. A sample is placed on the sample well. The sample pad receives the sample and acts as a filter to aid the flow. The conjugate pad contains gold conjugate antibodies that bind with their specific biomarkers from the sample and push them towards the nitrocellulose membrane by the capillary action. Nitrocellulose membrane contains test lines and control lines with capture reagents to display a result. No binding indicates a negative result, and visual control lines indicate that the test was run correctly. The labels give color to the control and the test lines. Then at the absorbent pad, it helps the sample flow and collect any excess sample. At the test line, the gold conjugate antibodies recognize its target and binding occurs. The test typically finishes in 5 to 10 minutes, where the presence of colored bands indicate positive results. Next, we will discuss glucose as part of the applications of microfluidics in blood analysis. For diabetic patients, the ability to rapidly and conveniently measure the glucose is critical for their diagnosis and monitoring. In-home glucose monitoring assays are available, and the finger-stick blood sample with a paper-based lateral flow immunoassay is used to quantify the blood glucose concentrations. However, more recently, there has been an effort to design less invasive continuous glucose monitoring platforms with technologies that eliminate the need for the finger prick blood sample. Examples of such technologies using microfluidics include the GlucoTrack, Pendra, and microneedle-based glucose monitoring. GlucoTrack is a device that contains an ear clip to measure glucose levels non-invasively via the earlobe. The reason behind selecting the ear as a site of monitoring is for being easily accessible with high blood supply. This table summarizes some information about the device. The technology employed uses ultrasound, electromagnetic, and heat capacity, and the advantages of using GlucoTrack include high precision and accuracy, calibration being valid for 1 month, having alerts for hypo and hyperglycemia, with multi-user support, data storage capacity and software for data analysis, the readings being unaffected by daily routine activities that the patient does, and having high accuracy in clinical trials with good correlation with glucose meters and glucose analyzers. The device itself is also compact and lightweight with large LCD screen. However, some disadvantages include requiring individual calibration every month and requiring improvements in the calibration procedure and algorithm for data processing. The second device is Pendra, which uses an impedance sensor that measures the electrical potential on the skin and the tissues underneath for signal acquisition and then signal processing, and then via an algorithm, it calculates the glucose levels and displays it on the watch. Lastly, microneedles-based continuous glucose monitoring, which uses hollow microneedles with microscopic channels to measure blood glucose levels non-invasively. The study uses 2 examples to compare the microneedle device, that is shown in blue line, versus reference blood glucose, that is shown in red dots, over a period of 72 hours. Results display good agreement in the glucose level readings for both devices. The researchers also plotted glucose levels when 2 microneedle devices were worn simultaneously by the same subject, and results confirmed the inter-device precision. We move on to discuss the applications of microfluidics in complete blood count (CBC). The complete blood count (CBC) can be used for multiple purposes. For example, we can detect abnormalities, it can help us diagnose various diseases, it can help us evaluate the overall health or rule out any disease or disorder. It can also help us monitor the progress of treatment. The Sysmex POC 100i automated hematology analyzer is a small footprint machine that only requires 15 microliters of whole blood to perform a CBC and a range of other clinical parameters, including a 3-part white blood cell analysis. This study demonstrated the use of a finger-stick blood sample to conduct a 3-part white blood cell differential using a microfluidic platform. The results table shows similar percentages detected for lymphocytes, monocytes, and granulocytes compared to a hospital laboratory analysis. The advantages of using microfluidics in hematology include enhanced sample control, the ability to handle small volumes, potentially faster time to results, analyzing small or difficult to obtain patient samples, especially with the pediatric patients that already have a smaller total blood volume and repeated sampling can be invasive. We move on to the final test exploited by this presentation, which is the sweat chloride test. Basically, this test, as the name indicates, measures the amount of chloride in the sweat. It is used to diagnose cystic fibrosis, which is a disease that causes mucus buildup in the lungs and other organs. It damages the lungs and makes it hard to breathe. People with cystic fibrosis have a high level of chloride in their sweat, and sweat samples must be taken from the skin of the patient, typically an infant, and sample volume is usually limited due to the low volume of sweat and evaporation. For that reason, microfluidics can have a great potential in this field. This figure shows a wearable skin patch for sweat chloride test. The skin adhesive is the layer in contact with the skin, and the patient's sweat will move through the patch towards the microfluidic channels, where the sweat volume will be measured alongside chloride and other electrolytes. Results will be visible in color references. To sum up our topic, we've given a brief overview about microfluidic systems, their fundamentals and physical properties, their fabrication method using photolithography and soft lithography techniques, the materials that are employed in manufacturing these small devices, and their applications in the nanomaterial synthesis and the hematology. Thank you for attending this presentation. I hope it was enjoyable to you as much as it was for me.